I'm just continuing the discussion we started last time of prokaryotic cell structure and function. And today we'll continue with some outer structures that are special. They're present in some bacteria, but not all, and their roles in the function of these bacteria, as well as move on to the characteristic structure and function of eukaryotic cells and some key differences between prokaryotic cell structure and function and eukaryotic cell structure. So some of these special structures that I want to bring to your attention are actually really important virulence factors because they enable the pathogenic forms of bacteria to attach and colonize our tissues as those pathogens that are human pathogens. So there are a few examples here that I give on this slide. One is pili, which is the plural version of pilus. These are appendages that are short. They're protruding from the surface of the bacterium, and they're used for attachments. They have special proteins that make up this polymer that is the structure of these tiny filaments that protrude from the surface. And those proteins are pilins, or in the case of fimbriae, which are very similar. They also have another type of protein that makes up their structure, but they have similar structure and function, pili and fimbriae. The pili have a special form, so sometimes they just coat the entire surface, and they're these kind of rather medium protrusions, not as long, certainly not as long as a flagellum, but they often coat the surface and help for attachment. They can also help for kind of gliding motility. And then there's one special type that some bacteria have and can express, and that's called a sexpilus. And that is a single long protrusion that can then attach and connect to other bacteria, not to host tissues, but to other bacteria. And through that, you could think of it as a channel or a funnel through which genetic information can be transferred directly from one bacterium to another. Hence the name sex, because it's a quicker way that genetic changes can occur as opposed to the usual asexual reproduction that is just an exact copy or near exact copy of the genome. Okay, so small chunks of DNA such as plasmid DNA can be transferred in its entirety through a sexpilus. Larger fragments or larger pieces of DNA like the chromosomal DNA would just be too much to this connection is temporary and the time is not long enough to mediate the transfer of an entire genome or entire chromosome. It's only these kind of smaller extra chromosomal pieces that get transferred by these special appendages. But we'll learn about that later when we talk about bacterial genetics. Fimbriae are similar to pili in their structure and function. They're actually physically shorter than pili, but they have the same function as far as attachment to a substrate such as a tissue, and they are primarily a polymer of protein monomers. And again, this is an important step in the pathogenesis for bacteria that are pathogens. In the case of human pathogens, they need to attach to our tissues before any colonization can occur. So these help in that first step of pathogenesis. And then there are also these interesting effects that once a pathogenic bacterium has attached, it can mediate effects on the of the cytoskeleton and change the shape such that the kind of protrusion on one end of the bacterium can form, and this is called a stalk, and it's where there's a tighter, more permanent attachment, more long-term attachment to host tissue, for example, that would occur. And the stalk then mediates changes in the host cell tissue such that it forms a pedestal for the bacterium to adhere within the tissue. And we can see this with many intestinal pathogens such as E. coli. And it's actually the factors that are secreted once that stalk forms. It's secreted at the area of the stalk into the host cell to which it's attached that mediate these changes in the host cell tissue. All right, so I did mention in the last slide that these fimbriae and pili are kind of like shorter versions of a flagellum, but unlike flagella, their primary function is for attachment. Only some limited gliding motility by pili can, if the pili coat the entire surface, can be mediated by those appendages. But in the case of flagella, or flagellum in case of the singular, if it just has one appendage, these are much longer appendages that their primary function is in motility. Okay, so usually motility is considered a virulence factor because it enables bacteria and other microorganisms that have flagella to move towards a source of food, so it enhances their chances of survival in the host, or away from a toxic substance. So again, enhancing their chances of survival. So it's considered a virulence factor, and we can measure the presence of flagella by special flagellar staining, as well as special biochemical tests for detecting motility that we'll be doing in lab. So flagella are actually molecular motors. By that I mean they're a set of proteins that use that convert potential energy into stored in the cell as far as the energy in the form of ATP into movement. And these motors spin in a certain direction, and so it gives this characteristic movement that bacteria have as a result of having flagella, where they have very intended directional movement, and then they stop when the flagella resets, and they tumble a little bit, and then they run in a specific direction. So run, tumble, run is very typical of bacteria that are motile due to the presence of a flagellum or many flagella. And the arrangement of these flagella varies depending on what type of bacteria you're talking about. They can be at the poles at either end of the cell, or at just one end, or completely coating the entire surface of the bacteria. In the case of many intestinal microorganisms, many intestinal bacteria are motile, whether they're normal microbiota or pathogenic microorganisms. And Proteus is one example that use flagella as part of their pathogenesis to swim up to the area of the bladder from the urethra, where they can infect either the bladder or swim even further up and infect the kidneys. So here's a transmission electron micrograph, and you can see that there are several appendages protruding from the cell, and those are flagella that coat the entire surface of the cell, similar to what you would see with Proteus species. And just as I mentioned before, the run and tumble motility is what the flagella will mediate because of their rotational movement. So when receptors on the surface of the cell, either in the cell wall or in the cell membrane, detect attractants, the flagella, or a flagellum, if it's only one appendage that the bacterium has, will all rotate in the same direction, and that direction is counterclockwise, which drives them in a run, specifically toward an attractant. And then they pause and reset by spinning in a clockwise rotation. That sends them kind of tumbling in random directions, but then with the next counterclockwise rotation, they again move toward the attractant. And so it's run, tumble, kind of zigzag motion, but eventually the net effect is to get towards the attractant, some kind of nutrient that they need or advantage to their growth and survival. And you can see that depicted here by the arrows. So directional movement in a run, and then kind of random movement in the tumble, and then again directional movement towards the attractant up here in the upper right corner. All right, so now I want to turn your attention to the eukaryotic cell. This should sound really familiar from what you learned in A&P or general biology. So I'm going to just suggest that you review this anatomy of the eukaryotic cell and all of these membrane-bound compartments, and just remind you that eukaryote means eu meaning true, karyo meaning nucleus. So it has a true membrane-bound nucleus. The nucleus is bound by a nuclear envelope, which is actually two lipid bilayers that surround the entire compartment. Other compartments within the cell include Golgi, ER, rough and smooth ER, lysosomes, peroxisomes, mitochondria. You could also have vacuoles and endosomes. And those compartments are all bound by a single lipid bilayer, similar to the plasma membrane or cell membrane. One organelle that is not membrane bound is ribosomes, but it's considered one of the organelles in any cell. So both prokaryotic cells and eukaryotic cells must contain ribosomes because they carry out the conversion of that RNA, the message that was formed from the DNA gene, and they convert that message into the form of a functional protein. Really the workers of the cell are the protein, and remember that's 15%, 15 to 16% of a cell, whether it's a prokaryotic cell having around 16% protein or a eukaryotic cell having around 15% protein. That's really, those are the key macromolecules that enable a cell to function and stay alive. So one thing that these internal membrane-bound compartments can do is they enable the cell to sample the environment around them, to bring in important particles such as food, or to internalize agents that are threatening in their environment in the case of phagocytosis, which we already went over in the end of Chapter 4 material. So this process is where, of endocytosis, is where the cell membrane here at the top of the image is able to change its shape once its receptors are bound by and recognize some food in their environment. And they can bind to that food particle, bring it in by changing the shape of the plasma membrane such that it inverts and then pinches off to form an endosome. And by the fusion of that endosome to a lysosome, the contents of that endosome can then, the food within the endosome can then be broken down and released across the membrane of the endosome into the cytosol, or if it's waste products, then it can just be released into the external environment. And this process of endocytosis is also what viruses, so these non-living agents, sometimes use to they kind of hijack this mechanism that naturally occurs in eukaryotic cells, such as our own human cells, in order to enter a cell and to start its infection and replication within that host cell. So there's also material that's being brought out. Let me just go back for a moment. From here at the bottom of the figure here, you can see the Golgi. So also products, proteins, toxins, etc., can be produced within the ER of a eukaryotic organism, such as a microorganism, and they mature through the Golgi and then are exit the cell by a secretory pathway, in which a similar process of the changing of the membrane of the Golgi complex allows vesicles to pinch off and then be shuttled out and fuse with the cell membrane and release their contents either onto the cell surface or outside of the cell. So the secretory pathway involves the rough ER, where the proteins are made, and then those secretory elements will then travel through the Golgi apparatus and to the surface of the cell. The smooth ER is where lipids are made. It also is used to kind of store or sequester substances that do not belong in the cytoplasm. And lysosomes, as I mentioned, are these compartments that fuse with endosomes to help break down their contents. And so an important feature of lysosomes is that they contain these enzymes, these protein factors that catalyze the breakdown. So they're lytic enzymes breaking down proteins, so proteolytic enzymes they often have a lower pH in lysosomes, so they can kill bacteria by causing uh, conditions in which the bacteria can't survive. So a lower pH, they start to degrade the proteins or the sugars that make up their cell wall and the proteins that make up important components within the cell wall and cell membrane, as well as internally. And also they deprive the bacteria of oxygen. So it's usually an anoxygenic factors that deprive that compartment of oxygen. So in the process of phagocytosis, when the phagolysosome, so the phagosome brings in the bacteria into the phagocytic cell, like a neutrophil or macrophage, and by the fusion of the lysosome with that phagosome is when the bacteria are killed and degraded. If that step is prevented, so the lysosome is not allowed to fuse with the phagosome, some pathogens are able to then survive within the phagocytic cell and continue to on their through their process of pathogenesis to cause infection and disease. So let's look for a moment at the organization of the nuclear material in eukaryotic cells. As I mentioned, there's a nucleus which is bound that contains all of the genetic information that's necessary to make a new organism. So all the chromosomes of that organism are contained within the nucleus. The nuclear envelope is two concentric phospholipid membranes, so we call it an envelope rather than a nuclear membrane because it has those two lipid bilayers, unlike other membranes within the endomembrane system. And unlike the cell membrane itself. And then within the nucleus, there's not a membrane-bound compartment, but there's a region of concentration where all of the RNA and protein parts necessary to make a functional ribosome are concentrated within the nucleus, and that's where the assembly of ribosomes, the large subunit and the small subunit of ribosomes occurs. Before they are transported out of the nucleus to the cytoplasm where they have their function. And here's on the lower left is just an electron micrograph that's been colored to highlight the nuclear compartment with blue. And you can see it's quite large in this cell type. It's usually one of the largest compartments in eukaryotic cells, with the exception of the vacuole in some cells, especially plant cells. And then within that compartment, the nucleolus is where the ribosomes are made. So remember, this is an important site, the nucleus within eukaryotic cells, where the DNA, the genes within that chromosomal DNA, are then converted to a messenger RNA. And then that messenger RNA exits the nucleus and through the nuclear pores, which are these darker purple holes in the nuclear envelope, and they enter the cytoplasm where they are taken up by ribosomes and either attached to the rough ER or are just in the cytoplasm itself, are converted to protein. Mitochondria, also part of those internal compartments that every eukaryotic cell has, but it's not part of the endomembrane system. It was thought to arise by symbiosis of a very ancient form of a eukaryotic cell engulfing bacterium and then their relationship becoming codependent. So this is what we have learned in previous classes as the powerhouse of the cell. It's where energy in the form of ATP is made because it's the location of oxidative respiration and the electron transport chain and the protein motive force that is required for the synthesis of ATP from ADP. It actually has, again, two phospholipid bilayers, so there's an outer membrane and an inner membrane. The inner membrane is this highly invaginated with lots of infoldings called cristae, which gives it a large surface area that it needs, where it can make, so that's the location of the electron transport chain, and it can, it can make a lot of ATP as a result of that large surface area, having many electron transport chains and many enzymes, the ATP synthase, which we learned about in Chapter 4, that are required to add a phosphate directly to the adenosine diphosphate to form adenosine triphosphate as a result of those protons moving down their gradient naturally, and that potential energy that the proton, the imbalance of the protons have across the inner membrane is really what drives that energy unfavorable reaction. And then in some eukaryotic cells, those that are capable of carrying out photosynthesis, they have another membrane-bound compartment that has actually the three membranes. So it's bound, so it has the chloroplast has outer membrane, an inner membrane, and a thylakoid space that's also membrane bound. So they are, these unusual organelles are responsible for converting light energy to produce ATP. So these are kind of self-feeding or self-sufficient organisms that can use light energy to make ATP rather than having to acquire that energy to make ATP from food, like carbon-containing compounds. And the thylakoid membranes are where both the light reaction and the dark reactions occur within the chloroplast, and the chlorophyll molecules within these thylakoid membranes are important for capturing that light energy. In addition to ATP, NADPH, which is an electron carrier, that are produced in the light-dependent reactions are then used to reduce CO2 to form food, sugars, in the light-independent or dark reactions. And we're not going to spend a lot of time learning about photosynthetic microorganisms since we're really focused on medical microbiology, and so pathogens are primarily, especially human pathogens, are primarily heterotrophs, chemoheterotrophs, so they require some outside carbon source. They cannot fix carbon to produce their own forms of food in the forms of carbohydrates or simple sugars. They have to acquire that from their outside environment. And our bodies provide plenty of food for them to do that. Finally, I wanted to touch upon a key difference. So I mentioned that both prokaryotic cells and eukaryotic cells have ribosomes because they're essential for making proteins, the working functional units of the cell that make up about 15 to 16% of any cell. But there's a key difference between prokaryotic form of ribosomes and the eukaryotic form of ribosomes. And this becomes important because while some antibiotics are very effective at targeting the cell wall and killing those bacteria, the gram-negative bacteria, which do not have a very thick many layers of peptidoglycan, are really not susceptible to cell wall destroying antibiotics, and so there are other antibiotics that are effective against them that target other structures within the cell, including the ribosomes. So in the case of the prokaryotic ribosome, they have both a large and a small subunit, just like the eukaryotic ones, but the sedimentation rate, which is a reflection of the structure and the both RNA and protein composition of these subunits is different. So we have a 30S. S is Fedberg units. That's just a measure of how it sediments when you put it in a gradient, in a thick like sucrose gradient, and spin it in a centrifuge. So it sediments differently than the large subunit of eukaryotic ribosomes, which sediments at 40S units. Same thing with a small subunit. It, in the case of prokaryotes, is a 50S subunit, and the total, if you don't separate the large and the small subunits and then you just sediment the entire intact ribosome, it would sediment at 70S. That's why these are not additive. Don't worry about that. So these numbers being different from the sedimentation rate of the eukaryotic ribosomes indicates that their structure, so their composition as far as the ribosomal RNA and the riboproteins that make up the large and small subunits, are really quite different. And as such, the binding of antibiotics will bind very well to prokaryotic ribosomes, but not to eukaryotic ribosomes. And so that selective toxicity can be achieved, and that we need when we're talking about effective chemotherapy for treatment of infections. Finally, I just wanted to touch upon some key differences between the eukaryotic cell wall and prokaryotic cell wall. So not all eukaryotic microorganisms have a cell wall. Remember that really it's just algae and fungi that out of the microorganisms that have a cell wall. Protozoa are defined as not having a cell wall. Helminths also don't have a cell wall. And importantly, as far as for selective toxicity of antimicrobial or antibiotic drugs, our own human cells lack a cell wall. So when we're looking at algae and fungi, though, the fungi have and algae both have a distinct composition of their cell wall that can, when compared to the cell wall of bacteria, which is much more complex, especially in the case of the gram-negatives, where you have the cell wall, but also an outer membrane in addition to the cell wall. And in the case of the gram-positives, where you have not just repeating chains of glycans, but also these peptide cross bridges that add the strength to that cell wall structure, to that peptidoglycan structure. So in the case of algae, they're made up of a polysaccharide called cellulose, similar to that of the plant cell walls. And in the case of fungi, they're made up of glycan chains that are polymerized into a compound called chitin, hard substance found in exoskeleton of insects. And I just wanted to also remind you that, as I mentioned, other microorganisms don't have a cell wall, but they still actually do have some rigidity to their cell structure and strength. That's in the case of amoeba and ciliates, they have a thick layer outside of the plasma membrane that's also flexible, because remember, amoeba are those microorganisms that engulf and ingest their food. And so it has to be flexible so that the cytoskeletal changes can be accommodated. So the pellicle is a thick layer, it's a protective layer, but it's not defining the shape of the microorganism. And instead of being composed of glycans or polysaccharides, It's composed of proteins that are found in microtubules. So a strong material, but also flexible material. And here's a table going over some key differences between prokaryote and eukaryotic cells. True for bacteria versus human cells, which is important to think about when we're trying to find drugs that will effectively treat infections by bacterial pathogens. So when we look at the size of the cell, we can right away under the microscope see that there's a large size difference. If we stain the cells, we will not see a nuclear membrane or nucleoli, whereas you will see that in staining a eukaryotic cell. So that's a reflection of the absence of any internal membranes internal to the cell membrane in prokaryotic cells like bacteria, whereas there are many internal membrane-bound compartments in eukaryotic cells. There is a functional difference in the protein subunits of the flagella in prokaryote versus eukaryotic cells, but I'm not going to expect you to remember that, and it's not relevant for treatment of these of bacterial pathogens, infections caused by bacterial pathogens. There is an important additional layer that we find in the form of capsules or slime layers that are glycocalyces that are present in prokaryotic cells but absent or in very rare cases are found in some cells that don't have a cell wall, so it's kind of a slippery, sugary coat that's only in some cells, not in human cells. And of course, in eukaryotic cells, like our own human cells, we don't have a cell wall, but some microorganisms do have a cell wall, and you should know which ones do and which ones don't. So bacteria, all bacteria and archaea have a cell wall, except for rare cases. In the case of mycoplasma, no cell wall. But the bacterial cell wall always includes peptidoglycan either in one or a few layers or many, many layers. That's not the composition of eukaryotic cell walls. They're chemically more simple. They include polymers of carbohydrates like cellulose or chitin. Cellulose in the case of algae and chitin in the case of fungi. There is also a difference in the types of lipids in a plasma membrane or cell membrane. So there are no carbohydrates in prokaryotic cell membrane, and they generally lack sterols, which are a kind of another kind of lipid like cholesterol, which we do actually in our human cells have plenty of cholesterol to help strengthen that structure, that outermost structure of our cells. And in the case of our cells as well as eukaryotic microorganisms, they all have carbohydrates that serve as receptors on their cell or plasma membrane. In the case of the cytoplasm, there's no cytoskeletal structures in prokaryotic cells, but clearly all eukaryotic cells have cytoskeletal elements like actin, microtubules, and also, let's see, actin, microtubules. There was one other thing I was going to say. Microfilaments, intermediate filaments, and these structures are as far as the cytoskeleton of the eukaryotic cells goes, help determine the shape when they don't have a cell wall to determine a shape. And I already went over the difference in the ribosomes that allows for us to have antibacterial chemicals that can target the bacterial ribosome and not affect the eukaryotic ribosome. Also, simpler chromosomal DNA, one usually just in the form of a single chromosome in the case of prokaryotes, and it is not tightly wound around proteins such as histones, whereas in eukaryotes, even eukaryotic microorganisms often have multiple chromosomes. They're linear in their form, and they're tightly wound around these complexes of proteins called histones. Then we'll get into, in our next chapter, in chapter 6, we'll get into more details of the cell division. But all bacteria carry out binary fission, whereas eukaryotic cells have, especially in the case of microorganisms, have a variety of ways that they carry out cell division, but it all is involving some form of mitosis. In most cases, some also carry out, some kind of yeasts also carry out fission, but usually the vast majority are carrying out some form of mitosis. And this binary fission form of cell division is an asexual form of reproduction that occurs in prokaryotic cells. The only way that they can really have a large change in their DNA is through this sex pilus that I mentioned, through a DNA transfer mechanism that we call conjugation that we'll learn about later in the semester. There is, in many cases of both microorganisms as well as our own cells, are able to carry out sexual recombination through the process of meiosis. All right, so I'm going to stop there. Here are some suggested practice problems for you to prepare for the exam. And that's all for today.